Hi there, Year 10. Mrs. Smith here, teaching you today about antibiotics. So the first thing I want you to do is I've sent you a task on Alexander Fleming. I would like you to use the 8 clue cards to try and fill in the blanks on a bit of a life history on Alexander Fleming. So if you haven't already done that, pause the video and do it now, and we'll go through the answers in just a second. Okay, Year 10, hopefully you really enjoyed the Alexander Fleming task. Let's go through what we need to know. So, the answers should be, hello, my name is Alexander Fleming, but my friends call me Alec. I was born in Scotland in 1881. When I was 33, I served in, now you can have 2 answers here, either World War I or the Royal Army Medical Corps as a doctor. While doing this, I noticed soldiers dying of sepsis. When the war ended, I started to look for ways of killing bacteria. I worked in London and studied bacteria. On the 28th of September 1928, I went into the lab as usual. I had left some bacteria growing overnight. One of my petri dishes was contaminated with a fungus. I was about to throw it away when I noticed the fungus. Around the fungus, there were no bacteria. They had all been killed. After careful study, I noticed that the fungus didn't kill all bacteria, only certain types. Even so, it was a big discovery. I gave it the name penicillin. However, it took me a long time and some help from Howard Florey and Ernst Chain before I could grow enough of it. We finally managed it in 1940 and were able to sell it in 1945. We were given a Nobel Prize for this work in 1945. My advice on taking antibiotics is the same as it was when penicillin had just been discovered. Take the full dose and don't stop taking it early. Some people don't follow that advice, and that's why we have antibiotic-resistant bacteria. And so that's how antibiotics were developed and discovered, which is actually quite exciting, and I hope you enjoyed doing that task. So, what antibiotics do? Okay, anti means against, and bios means life. So you put them together, antibios, you get against life. Okay, so they're things that kill off life forms, particularly bacteria. And there are lots of different antibiotics, not just penicillin, and they all kill slightly different bacteria. Okay, it's just like when we looked at the immune system and you need certain antibodies to kill off various different types of pathogen. You need different antibiotics to kill off various different bacteria. They only kill bacteria as well. They won't kill fungus, they won't kill yeast, and they certainly won't kill viruses because viruses aren't alive. Okay, so we've got antibiotics, but people don't take the full course. They stop taking them, or they don't complete the course, or they stop early. And that means if there's any bacteria left in your system, those bacteria can multiply. And when there's, so you take your first few antibiotics and they kill off most of the bacteria. And then you take a few more antibiotics and they just beat down and end up killing the bacteria. However, if you stop early, then those bacteria that haven't quite been destroyed by the antibiotic are like, we fought it off once. That's how we do it again, just like your immune system. Okay? So the antibiotics become useless because the bacteria know how to fight them. It's why we get all these resistant bacteria. So you've heard of things like MRSA, which is a bacteria that normally lives on your skin. It's absolutely fine, but if it gets into a wound, it can give you blood sepsis and kill you because antibiotics don't work against it. So what can we do? Well, the good news is that we can develop new antibiotics and new drugs against these things. But if we're going to do that, we need to test the drugs. Because if I came up to you and said, yeah, all right, got this drug here, think it might work, want to try it? Hopefully you're going to say, no, Mrs. Smith, I do not. So we need to find out how we can make drugs such as antibiotics, but also any drug that's going to go onto the market to do anything. What do we have to do before we can use that drug, or indeed vaccine? Because obviously there's a lot of talks about developing a vaccine for COVID-19 at the moment. So why is it taking so long? So we're going to find out. I'm going to use the book to help me. So let's take a look. Okay, so this is what's needed in developing a new drug. First of all is preclinical development, and these are the tests in the laboratory. You run simulations on a computer and in test tubes. Sometimes for some drugs, you need to see how it's going to work on a full living organism, and so you will do animal tests. Very few drugs will be approved for use in humans if it hasn't been shown that they don't cause harm in animals. That's just the way the law is at the moment. But most of it these days can be done with simulations and test tubes, so animal tests are being used less and less frequently. Once we've figured out how the drug should work chemically, and checked that it works against the target molecule in a test tube, then we can move, or maybe in an animal, then we can move on to the clinical trials. And there are 3 phases of clinical trials. Can I just point out the preclinical development bit at the top? That can take 3 or 4 years to do. So clinical trials, first of all, you round up about 10 or 20 perfectly healthy volunteers and you give them a fitness test. You make sure that everything's fine. They've not got any underlying health problems. And the reason you need to do that is that basically if anything in this drug makes you ill, then we want to know that it is an effect of the drug and not an effect of the illness that you already have. So if, for example, I wanted to test a drug that should lower my blood pressure, for example, I'd want to test it in healthy people to make sure that it did lower the blood pressure, because if it increases the blood pressure, then it's really not a good one to give to people with high blood pressure. Or if it made people violently ill, I'd want to know that it was the drug that was making them violently ill and not it mixing with an underlying health condition or another drug that they were taking. This, of course, can go wrong. So if the healthy volunteer took a drug to lower their blood pressure might go a bit too low, they might feel faint, they might collapse. There is a risk of death with this. There was one less than 10 years ago where they did a clinical trial, and instead of doing 1 patient at a time for 10 patients, they did 10 at once, and these people ended up with leprosy-like symptoms, I think it was, and they basically, their fingers and toes all died, and a lot of them needed to be amputated. So it's not without risk. That's why these healthy volunteers are often paid. These healthy volunteers are often young men as well. This means that drugs haven't been trialled in women yet, only in men. And of course, drugs can have a different effect because we've got different levels of hormones in our body. There are some people saying that this is a problem, and indeed it is, and we do need to do more research on that. So clinical trials 2, let's move on, is your target patient group, and you take a small amount. You're looking at maybe 100 patients, and you say, we think we've got a new wonder drug, and we think it's going to do more good than harm, and we think it's the best treatment for you. It's better than what's currently out there. Would you like to try it? And you as a patient can say, no, I don't want to. Once you've got this small group, you check that the drug actually works and it doesn't have any side effects and it does what you think it's going to do. So if you give it to people with high blood pressure, does it lower their blood pressure? Once that's gone through safely And these tests can take a good year or so each. Then you do a larger target group, and this can take a couple of years at least, clinical trial 3. And you've got a few thousand people. And if you don't do it all at once, you might do 200 at a time for 6 months, which is why it takes a few years. And you will do, say, 200 people, and you will offer them a new drug, but they won't know if they've got it or not. That's what a placebo is. A placebo is a placeholder. It's something that people often call them sugar pills, but that's not true anymore. It's usually a continuation of the same medicine so that you can see if the new drug has a better or worse effect than the existing drug, because you don't want to put something on the market that's worse than what's already out there. So you will check in a larger group what happens if you take the drug, what happens if you take your normal drug. Is the new drug better? Is the new drug worse? Which one has the worst side effects and things like that? Once that's been done, then it goes for review. And again, this can take up to a year. And all the risks that you found out in your clinical trials are totted up, and they say, right, okay, so our blood pressure drug has the risk of, it makes you more tired and it's making people overeat. But the benefit is that it works twice as quickly as previous ones, and people don't seem to get the headaches, and they don't seem to lose their sense of smell. I don't know, just making that one up there. So if it passes review, if it's deemed as being safe based on the clinical trials and better than an alternative, then it's launched. And at drug launch, that's it. It's rolled out. The NHS can prescribe it, or whoever else can prescribe it. But this is the important point here. It is still studied. Okay, so if you open up a packet of paracetamol, for example, and it says, these are the likely side effects. If you take paracetamol, then it should get rid of your headache, but you might feel a bit nauseous, for example. Some people say that they get tingling in their arms and legs. I don't know, I've not read a paracetamol box for ages. You should do it every time you get a new box of drugs, by the way. So I've obviously not taken paracetamol for a while. If you do get any side effects, which you are pretty certain relate to the fact that you've taken that drug. So if I took paracetamol and then my eyes stopped working and I went blind, I'd be like, okay, I better call the number on that sheet somehow, not quite sure how now, I can't read, and because I'm blind. Or you'd call your doctor, and your doctor would log the fact that that drug had possibly had that side effect. And if it has that side effect for a few people, then it goes onto the leaflet as a rare side effect so that people are aware of the risk of taking the drug. One final thing before I go is that another reason to study it is because it can have different effects. Now, most of the clinical trials are done on men because apparently women's hormones changes, so it's too difficult to look at unless it's a drug marketed at women. So most of the trials are done in men. It can affect women's bodies differently, and it can certainly affect pregnant women differently, and it can affect the developing fetus, especially in the first trimester. There is a drug called thalidomide, which I'm not going to go into here too much, but it was given because it had the side effect of stopping morning sickness, but it interfered with the development of the fetus, and the fetus wouldn't grow limbs, so you had babies born without arms and legs, and it took them years to link this, which is absolutely dreadful. But it's now found, so long as you don't give it to pregnant women, it's a really good treatment for leprosy. When I was pregnant, we had to discuss, do I take some drugs to look after the problems I was having in pregnancy, the medical problems, Or is it safer not to? Because you can't do a trial in a pregnant woman. It is just not ethical because you don't know the effect it's going to have on the fetus. So the doctors could tell me they've prescribed it to pregnant women before and it hadn't been a problem. And we'd had a scan and my baby's limbs had all developed and their organs had all developed already. So the risk was minimal, but the risk to me of not taking it was greater. So we had to discuss that and decide, and that's why we still do all these studies. This is why we still have to study it so that we can say it might not be 100% safe, but it is safer than not taking it. Or if it does all of a sudden, maybe if you've been taking it for 20 years, you take a drug for 20 years, all your hair falls out, and you have a heart attack. And the next person who's been taking it for 10 years, all the hair falls out, and a week later they have a heart attack. Then someone else, and by the time you've had like your third patient come in and go, doctor, I've been taking this drug for 10 years and all my hair fell out. You're like, stop taking the drug. I don't want you to have a heart attack. So that's why we've got to study it, because there might be long-term effects that we didn't know about. And that's it for how to develop a new drug. That's it. That's all the information I need you to know about antibiotics and drug development. And that's also it for topic biology 5. That's it for paper 1. So we've done all the biology we needed to get done this year at school. Well done, you guys, for working so hard in lockdown so that we have been able to get through all this. Okay, I really mean that. I think you've done really well. Those of you who have been emailing me work every lesson, every week, I really appreciate the effort you've put in. Those of you who've been doing it most weeks, brilliant, you've been doing it. Those of you who've been doing it less often, you might want to review some of the videos because we will be doing a test. Don't worry, I'm going to be revising with you. So next week I'm going to be revising. If there is anything you particularly want me to go through that we've done in topic 5, which, if I just check my book, that's all of the diseasey sort of stuff. So that's health and disease, non-communicable diseases, cardiovascular disease, infectious diseases, spreading pathogens, physical and chemical barriers, the immune system, and then today's antibiotics and drug development. If there's anything in particular from any of those lessons you want me to go through, let me know. Otherwise, I'll be doing 2 revision lessons because there's 2 parts that you need to do for revision. And I'll deal with that one next week because this video is quite long. You're probably bored of my voice. So you've got another worksheet to do. Off you go, do that. It's the last worksheet that I'm going to give you, the last full-on lesson and worksheet thing we're going to do for biology. We've still got some more for chemistry. And yeah, well done. Just seriously, well done for getting to the end of this. Take care, and I'll see you soon. Bye.